It's my privilege to introduce Sergey. He has been, in many ways, a pioneer in developing next generation models for protein sequence, structure, and function. And today he's going to help us venture into interpreting these models, which, as we've seen, is quite important to understand what they're actually learning. And so look forward to his talk. Thanks, Sergey.
Thank you. Thanks, you guys, for inviting me to be here. It's really great to be back. I think first time I came to this conference actually was still part of NeurIPS, so it's been interesting to see the evolution. So today I'll tell you guys about protein language models, but since a lot of us care about genomes, I threw a little bit of genomics in there as well. So I'll tell you guys a little bit about some of the investigations that we've been doing trying to understand what different protein language models are doing. Some of these slides may be familiar to you guys because some of my past collaborators have actually presented at this conference before. So, but maybe this will be a refresher for those of you who've seen it before, maybe for new people also as well. All right, so the big picture or the roadmap that I'll follow today is quickly introduce you guys what are sequence models, or at least the ones that we use, how these relate to protein language models, and how different are they. And then towards the end, describing what's also been learned about genomic language models, and potentially how these two things are connected, and how tokenization takes a role in what these models end up learning at the end of the day. So big picture, let's say if we have a sequence, and the sequence could be DNA sequence, RNA sequence, or protein sequence, and we have one sequence, there's not too much we can say about it. But where things become really, really powerful is when you actually have a collection of sequences, right? So you have one sequence and you collect a whole bunch of sequences that are related, and now this becomes really, really powerful because now you can say, hey, this is like a very conserved position, these may be covariant positions, maybe this position is completely random. So it's through power of comparison that things become really, really powerful. But how do you actually make these comparisons? Like what kind of model could do these comparisons for us? And so some of these comparisons and some of the things that you could learn from these models is like, for example, like as I mentioned, conservation, phylogeny, coevolution. And one example that I'll be focusing on today is this idea that one really, really powerful thing that you can extract from these kind of data are these coevolution signals. And the reason why it's quite important to get the signal is because let's say if you want to predict the structure, using this coevolution signal, you could potentially tell you like these two positions are close together in the structure. And so you guys may have all heard about AlphaFold. At the end of the day, it looks like AlphaFold is essentially still using this exact same information to make the predictions. It's extracting these covariance patterns from a collection of related sequences, and that's how it works. And what I'll try to argue today is that even these new language models, protein language models have been trained, are essentially still doing this same thing, but less implicitly, less explicitly, more implicitly. Okay, so how do you actually model these constraints? So I'll just walk you through a toy example. So let's imagine we have a multiple sequence alignment, a bunch of sequences. Two positions are covariant with each other, and one position is completely conserved. I'm going to kind of take that one sequence, let's say from human, one hot encode that sequence. I just need to describe it somehow in order to pass it to a computer program. So like for example, blue was 1, 0, 0, and so on. Then I could come up with some kind of a neural network or a layer that describes the relationships. You can think of it as like training some kind of graph model, or you could just think of it as a dense layer that connects everything to everything. So for example, like position 1 is connected to position 2 is connected to position 1, and so you could essentially use a very simple matrix to describe these relationships. And then finally, I know some positions are completely conserved, like sort of in my toy example, like this guy's conserved, it's not dependent on anybody, so I can add some kind of bias term to describe that at the very end. And now finally, let's say if I wanted to train a model, essentially like an autoencoder that goes input to output, I have to make sure their outputs sum up to 1. And so here I could add some kind of softmax activation to make sure every position sums up to probability of 1. And then finally, I could try to train this model to essentially recover the inputs on the other side. And if you imagine training such a model, you'll probably learn something very, very simple. Like in this case, it could just learn an identity mapping. And so to get around this problem, what people in practice do is employ pseudo-likelihood. And so in this case, pseudo-likelihood would essentially be saying, let's just mask the diagonal, make sure that there's no self-connections. So the idea is like, let's say if I want to know how 1 depends on 2 and how 2 depends on 1, if I remove connections with yourself, then instead the only way to explain that would be to pass through another residue through a different position. And so now we can actually learn these weights would represent those connections. So this is just history of how we typically used to do this. And one particular example of this model is a method called Gremlin, and this is the method I'll be comparing to as far as comparing language models to these traditional methods. And the reason why it's called Gremlin is because you're regularizing it, and it turns out when you do name your algorithm, it doesn't always have to be the first letter of each word. It turns out you can use any of the letters. Just heads up if you want to be more creative in your names. Yeah, so, but the main thing here is that it's essentially the same model, that just what I described to you, but sometimes it's easier to think of it as a neural network as opposed to some kind of graph model or Markov random field or a POTS model. It's essentially just a single layer, and you're just doing softmax and then minimizing the cross entropy between the inputs and the outputs. But once you've got these parameters of this model, like what do you do with these parameters? How do you even extract these contacts that I just told you about? So what you could do now is you could take these weights and you get back some kind of tensor that's length by length, in this case alphabet by alphabet. And then you could take each of these little boxes here that's alphabet by alphabet and reduce it to a single value. So usually you take like L2 norm or a Frobenius norm, and you get back a contact map. And so now the question is like, okay, that looks pretty cool. And just to summarize, so now we train it on this data, and now we have essentially a contact map that represents the connectivity or how the different positions are dependent on each other. So this is just for a quick toy example, and just to show you that I'm not making things up. Here's an actual protein. So here you can see in gray are actual contacts coming from the crystal structure. And this is one particular example of a protein. This is a ribosomal protein. And let's say if I wanted to show how these these cyan contacts interact with these orange contacts, I could just look it up here. So cyan, orange, that's they are. So that's how you interpret contact map. I'll be showing quite a few of these, so I thought I'd spend a little time showing you how to read these guys. But now what I could do is I can overlay these contacts that I get back from this essentially a covariance matrix of this multiple sequence alignment, and you could see they actually overlay really nicely. So this is actually a really, really exciting result back in around 2009, 2011, where people started realizing, hey, these are actually really, really powerful information just by simply mapping this very, very simple model, or training this very, very simple model. And this sort of allows you to imagine a scenario where, let's say if you didn't know what the structure was, let's say if it was just some long extended chain and you put like rubber bands everywhere, you could potentially start to snap things into place. Like all these white lines are positions that are covariant with each other, and now you could potentially build a structure that maps those contacts. All right, so how about protein language models? How are those connected, and is there any difference? Are they the same? And so what I'll try to argue today is essentially it's almost doing the same thing. And what I mean by that is you still have an input sequence that you're one-hot encoding, you're passing it through some kind of layer, and you're returning probabilities at every position, the probability of that amino acid condition on everybody else. And in this case, and let me just break this down a little bit further, just to make it more interesting. But before I do that, just to quickly remind you guys the big difference between the two ways of training. So imagine with methods like Gremlin, what we usually do is we train one model per protein family. So for example, you have a ATP synthase, you have a separate model. Beta-lactamase, you have a separate model. With methods like protein language model, you usually train one model for all sequences. And usually if you do have to do that, then you might have to also think about positional encoding, because every protein might have some kind of different meaning at every position. You maybe have to do some kind of relative positional encoding, because maybe you want to allow for things to move around, like have insertions, deletions. And so, like in this case, it's a very, very simple model, but in this case, you do really have to pay attention to how you actually encode the positional information into the model, so it actually learns something useful. Okay, so let's break it down a bit. So let's say we have a sequence, which I'm just going to show as a gray box for now. I could pass it through a very simple linear layer and also add some kind of positional embedding. And so this is my new inputs. Then I could pass it through 3 more layers to get query keys and values. And if I take the outer product of these keys and values, I can get essentially how each key attends to each query. This is also referred to as an attention matrix, for those of you familiar. And then once you have this attention matrix, you now have to populate those attention with some kind of values. And so you multiply by the values, and you get a new output. Now, once you have this new output, you could pass it through another layer, which in this case I'm just going to call a decoder layer, to get back the sequence. And then you could take the softmax of that sequence and minimize the difference between the input and output. So essentially, it's the exact same objective as the Gremlin model that I just told you about. But in reality, what happens is, here I'm just showing it for like a single layer, a single head. But in reality, you could go a bit further. Like you could say, hey, there could be multiple heads. And so, but one thing that I should probably emphasize here is, in the case of Gremlin, it was really, really easy to fix the problem of information just coming right through the model, right? So like one question you might wonder, like, why can't the model just learn an identity matrix once again, right? And so to get around this problem, often people, what they do is to train these models, they would train it with a masked language modeling objective. And so what I mean by that is take your sequence, mask some positions, and then in this case, you only want to predict those positions that you masked. And so what I'm trying to argue in this particular slide here is that the pseudo-likelihood objective is actually very, very similar to the masked language modeling objective. In that case, you're masking some positions and recreating them. And in fact, what you could do technically is you can go to every position one by one and mask them one by one, but that would be really, really slow. And so in practice, people mask like 15% of the sequence. But in some ways, the two functions are approximating each other. So just based on this information alone, one could probably argue, like, okay, maybe they're learning something similar, right? Okay. All right. Oh, I don't know if anybody has any questions, but if anybody does, feel free to ask during. I don't know if we have to wait till the end. Yeah. Any questions before I keep going? Or concerns? Otherwise, I think I might be going a little too fast. So I feel like somebody has to slow me down a bit. Okay, so all right, so next, oh yeah, go ahead.
I guess you mentioned briefly that the one-hot encodings were dependent, they were different length depending on the length of the protein sequence, but somewhere we got around that, I missed it. At some point they all become the same length. Is that correct or no?
Great question. So here, when I'm showing these little arrows going from sequence to input, or even from input to keys, you can almost think of it as just doing like a 1D convolution. You essentially apply the same weights to every position the same way. And so you might imagine, like, oh yeah, that seems kind of silly. If you apply the same weights to every position, it would probably don't matter how you rotate your sequence, how you shuffle your sequence, right? And so to get around that problem, you add some kind of positional embedding. And this positional embedding could be like some kind of sine wave, or it could be a combination of sine and cosine waves. And essentially what you want to do is essentially give every position some kind of unique representation. It's like position 1 has this representation, position 2 has this representation. In practice, for protein language models, what's found to be the best is to do something called rope embedding. What rope embedding essentially at the end of the day does is learn some kind of just relative representation. So I guess you could say, like position 1 and position 2 are going to be really different from each other, but position 2 and 3 are going to also be relatively similar to each other. And so you essentially just encode some kind of relative embedding instead. And so then what this allows you to do is now if you give it a protein of different lengths that is the same, you essentially still have similar relative encoding of that sequence. And so then you can get away from the problem having to align those sequences. Now, if they're all aligned, your positional embedding could literally just be an identity matrix that literally says this is position 1, this is position 2, this is position 3. But if they're not aligned, then you have to provide some kind of relative information. Some of the earlier models, like the tape model, actually had a problem where they didn't do that. They essentially had some absolute positional embedding. And in that case, what happened is like you literally shift the sequence by one position, and it just crashes, no longer works. So it literally learned the perfect embedding for every sequence, but then it was not able to generalize as soon as you added like one position anywhere in the sequence. So this becomes really, really important on how that works. Yep. All right, so, all right, okay, in that case, I'll keep going for now. All right, so I just described to you guys how you go about training such a model, and I guess the argument I was trying to make is that it's the same inputs and the same outputs. Even the loss function is essentially approximating the same thing. And so maybe a spoiler, it may be potentially doing something very, very similar. But how do we actually demonstrate that it's doing something similar? And so one approach we took is that, well, what if we just train a protein language model from scratch and on a single protein family that's already, all the sequences are aligned to each other? What will happen if we just train a single model on a single protein family? Will it actually learn something interesting? Will it learn the same thing or not? Because here we have full control over the inputs we give it, and that's the only thing that it sees is the same exact inputs. And so what we found is if you train it on a single protein family, you actually only need a single layer to essentially return the same results. The other thing that's kind of interesting is because if the sequences are aligned, like I mentioned earlier to Jeff, is that you could essentially put in the positional information could literally just be a one-hot matrix. which specify, or I guess you say identity matrix, which specifies position 1 is 1, position 2 is 2, position 3 is 3, and so on. And so it actually becomes really simple model. But now the really remarkable thing, which probably is not too surprising in hindsight, is if you extract the querying keys, like the actual parameters of the model that represent the queries and keys, and take the outer product of those guys, you actually get back on contact map. And so within the parameters themselves of this very simple protein language model are already embedded all the same parameters that you get back from, let's say, like a gremlin model. And just to make things more confusing, turns out you can, I guess you could say, refactor a simple attention model into different components that essentially connects all the parts. And so the reason why I'm showing this is, like, for example, in a gremlin model, we have a weights matrix. And that weights matrix could essentially be decomposed into two matrices, where you say, hey, I'm going to model all the interactions and have some kind of heads parameter. And then each heads might be representing some 20 by 20 matrix describing how those parameters interact with each other. You could further decompose that into, like, this parameter could be decomposed into queries and keys. This could be continue going on. And eventually, you could see the values and the final feed-forward network are actually going to tell you all the information about how they're interacting with each other. And then the queries and keys are essentially the decomposition of this length by length matrix. And so for a single protein family, you essentially could go from beginning to end, like fairly completely decomposing it into the query keys and values and the final feed-forward network. Or you could just keep bringing it all together back into a single linear model that just has the length by alphabet matrix. Now, if we actually go through the process, because now that we know how the relationship between this and the queries and keys and values, we can actually now take the queries and keys and values and compare it back to this matrix and see, are they actually learning the same thing, right? And so I guess the argument here is technically if you make this H, which is like the heads, large enough, it should actually be able to reconstruct this matrix exactly. And that's essentially what we see. So here I'm comparing this W matrix that we get from Gremlin, but also this W matrix that we could pull out of a protein language model. And what we find is as you increase the number of heads, those parameters become more and more equal, which I guess mathematically kind of makes sense, but it's kind of cool to see that it's actually essentially, if you make heads the largest possible amount, which I guess in this case would be like 400 values, then it essentially should start equaling each other. Okay. So, but this is just for a single protein, single protein model. For a single protein family. What if you actually train this model on all the sequences, right? And so this is actually in practice what people do. People don't train a separate protein language model for every protein family. They train one model for all proteins. And so when you do that, often a single layer is not enough. Like, you have to actually have multiple layers in order to get a meaningful pseudo-likelihoods from the model. And so in practice, you have multiple layers. And in fact, often you have to have up to like 33 of these layers. And so just to give you an example, and one way we could potentially interpret these models, if you take a sequence, pass it through many of these layers, you can start looking at these individual layers one by one. And it turns out if you start looking at these layers, they actually capture some really interesting features. Like some of the earlier layers, very similar to some of the early work on convolutional neural networks, are kind of capturing some low-level features. But as you keep going to the end, it turns out often the last layers are beginning to capture these contact maps. And so some of the hypotheses are maybe like some of the first layers maybe picking up some local structure, and then as you keep going further, you end up building up the entire contact map. And this is an analysis of across all layers, across all heads, and what I'm showing here is the data points that are showing how accurate these heads correlate to contacts. So the higher you are, the higher correlation you get. And so you should see, the interesting thing you see is often towards the end is when you start to recover all the contacts in the model. Besides us, there's been other people that showed similar things. One paper being Biotology Meets Biology. So if you guys are curious, you can check out some of that more work. But one thing that I wanted to sort of highlight is if you start to actually compare the performance of these models, like the performance of how well these contact maps correlate to ground truth, they depend highly on the number of training examples. And what I mean by that is if you take a protein family and you ask the question, or you take a protein sequence and you ask the question, how many examples are there in the training set that are very similar to that protein? And there are some proteins that don't have that many examples, and there are other proteins that have a lot of examples. And so it turns out with gremlin models, we saw that before where the correlation is really rapid or striking, where like the more data you have, the more accurate these models are fit. But the interesting thing was even with these language models, you see the same kind of correlations, where the more examples there are in the training set, the more accurate these models become. But one thing I should emphasize when I say training set is like we're not training the models to return any context, purely unsupervised learning, but just the parameters themselves have better correlations to contacts. And ESM2 recently did the same experiment again, but one thing they had access to, which we didn't at the time, was this ability to say, well, we could train models larger and larger and larger, right? So in this case, I'm comparing it to one of the initial tape models, ProteBERT, and so on. But what the ESM team group did, and I believe Alex Rivas may have talked about this last year at this conference, is that the idea is that as you increase the number of parameters, you actually start getting better and better contact accuracy. But one thing I wanted to highlight here is this orange line here. So this orange line represents examples of proteins that don't have that many examples in training set. And as you see, as you start to increase the number of parameters, it does indeed start to get better and better and better, but it has not yet reached this level here. And I think at some point I was trying to extrapolate how many parameters you would actually need to get there. And I think the estimate right now is 10 to the power 25. So you need to burn a lot of trees to get there, I guess. But eventually we'll get there. But that's assuming that it's linear. Who knows? It's probably not going to be actually like that. Yeah. Oh, yeah. Question? Yes. Yeah. Uh-huh.
So is it the amount of training data, or is it actually the unbalanced sampling? So like what would happen, is it really that you just need more training data, or is it that you're seeing more of one type of family versus, so what if you upsampled or did some kind of balanced sampling of families? Would that help, or does it not help at all?
That's a great question. So in some ways, there has been some data balancing in terms of, like during training, often what people do is they first cluster all the sequence data, and then they pick one member per cluster when during training. So there is some degree of data balancing going on. But one other hypothesis was like, maybe we just don't have enough capacity, right? So one of the things that Alex Rivas' team says, this is why they're calling it evolutionary scaling. They're like, okay, we just need to scale, and if we scale, we should potentially be able to, even for the very, very low protein families that don't have that many sequences, eventually they'll be able to also capture those parameters as well. But I guess what I would argue is even if you have infinitely large model, eventually things start to plateau, and that's essentially what we're seeing with this orange line here. So those very small protein families, they are now getting better and better in terms of their, the model seems to understand those small protein families really well, but even at 15 billion parameter model, it's not getting much better. This is just like wishful thinking, say, well, if you make the model infinitely large, maybe eventually it'll get there, but in reality, I think it will essentially just plateau. Now, there is some hope saying, well, with all the metagenomic sequencing out there, maybe we'll start to sample some of those families better and better. But I don't think that will solve the problems for like eukaryotic protein families, for example. Like eukaryotes on average are very, very similar on the protein level. And so I don't think it's going to be any kind of, one thing sort of related to this plot, sometimes people ask me like, how do you know that the model has now had some kind of a breakthrough? I don't know if AGI would be the right word for this, but I would say if indeed you see like this line is flat, then I would say, okay, maybe the model has actually learned some kind of protein folding, maybe the model has learned physics, because the idea is like you give it a sequence it's never seen before, it should be able to understand the structure of that sequence really well. But in reality, what we see is this really, really strong correlation. And we do think it's one of those things where it's not because of data balancing, it's more like there's just, I mean, it's probably a little bit of both, because in some ways, just there's not that many examples for that, and there's no reason for it to even store that information to that family. But even if it had the ability to store it, there's like nothing to store, because there's essentially all those sequences are very similar, or just a few of those guys. Yeah. All right. Okay. Any other questions? Yeah. Oh, I think there's one all the way in the back.
Hey, so do you already see that double descent slope in these models at that plateau, or is it like all dependent on protein families?
So this is showing an average across all protein families. So you could think of it like this line represents protein families that don't have that many sequences, and this represents protein families that have a lot of sequences. And if we just focus on protein families that have very low number of sequences, you see that it just sort of plateaus. And I mean, I guess there is some hope. Like I guess this is where I was trying to draw this line here. But I don't think it's gonna. I don't know if that was the question, but I don't think it's gonna get any better for those families, unless it does somehow snap out of just the effect of learning conservation coevolution about these guys. Yep.
Yeah, so do we accept that maybe a protein structure might not be enough? So maybe there's some interacting proteins that help to stabilize the structure. What are some thoughts about that? And if you fold in some information about interacting, like known protein interactions, could you improve performance?
It's a great question. So in this case, the model never sees any protein structure at all. It only sees sequences. One thing I'll talk about a little later today is there are other models that you can give it pairs of proteins or more than just a single protein. In there, I guess, I mean, we would expect maybe to see improvement in prediction between the proteins, but maybe not so much within the proteins. But if it does improve something within, that would be actually kind of cool. But that's not something we looked at yet.
So one more question along the previous line. So if you take a look at these proteins that have very few members, I mean, the performance is not zero. It is slightly better than random. So what's your interpretation? Is it actually learning some physics, or are we underestimating the number of real neighbors?
It's a great question. So I don't have a line here, but it turns out if you actually just make a very, very simple model, say, given the amino acid identity, what's the probability that it'll be in contact? It's actually around 20% or so. So it's actually not too bad. And so I would say, I mean, Gremlin, this model knows nothing about proteins, right? There's no priors about proteins. So at bare minimum, maybe some of these jumps, for example, above 1 might just be coming from the fact that you say, hey, for example, one thing that you're adding is positional information. Gremlin knows nothing about position. And so you might say, hey, if you're 5 residues apart, you're more likely to be in contact than if you're 30 residues apart. So a lot of this is actually just coming from those very simple statistics that you could have probably added yourself. All right, so given all of these results, what this sort of tells us or leads us to suspect is at the end of the day, protein language models are essentially just learning a library of Gremlin models. So this is a visual here saying, given a sequence, maybe somewhere inside the parameters of this model, it's looking up the Gremlin model, and then based on that Gremlin model, it's returning to you the probabilities and statistics for that protein family. And so another way to maybe reframe protein language models is some kind of fancy lookup and compression algorithm. But that being said, there's a chance it might do better because of shared parameters. Because right now, when we do fit these individual models, we're only providing information for that one single protein family. But maybe there's multiple protein families that have similar kind of motifs, and those motifs could potentially help each other in some ways. So we do expect that they will do better than just training separate models. But at the end of the day, it's still essentially compressing, or at least this is what we believe they're doing. And so I guess let's say the 3 hypotheses about how the protein language models are storing structure is protein folding, which I already told you is probably not the case. But the 2 cases that I'll talk about for the rest of the talk are these: is it looking up entire proteins, or is it maybe looking up little motifs? Because instead of storing individual little models for each protein family, you can imagine it's storing like common motifs that are shared across proteins instead of storing a separate model for each protein family. And so I guess I'm just going to rule out this one for now. Say, okay, it's not doing protein folding, because if it was, then we would have expected this line to be flat. Like, it shouldn't be dependent on the number of sequences, right? So that are already telling us that's probably not the case. Maybe with a 100 billion parameter model, maybe we'll get there, but who knows? We'll have to see once we have enough GPU power. Okay, so we're ruling out this for now. So now that leaves us with 2 different hypotheses, that it's looking up entire proteins or it's just looking up motifs. But before we can even go about answer that question, how do we actually go about extracting information from these models? So I told you guys about extracting contacts themselves through the attention matrices, and sometimes this is not possible, like especially some of these newer models that use like these hyena layers, you don't actually have explicit attention layers that you could look at. But in other cases, like if we actually want to argue that it's learning coevolution or storing coevolution, we want to actually pull out the exact dependencies, not just some approximation of dependencies. So how do you pull out dependencies from these models? It turns out one way, a really relatively straightforward way, is to say, well, what if we just map every input to every output? Like just essentially create a matrix that shows how every perturbation of the input affects the perturbation of the outputs. And one particular example of this is to compute like a Jacobian matrix. So like, for example, if you take a model like Gremlin, which is a linear model, and you compute a Jacobian, which essentially is a matrix of partial derivatives. It's essentially telling you how every input, how perturbation of the input affects the outputs. You get actually back a really nice contact map. If you do the same exact thing to protein language models, it becomes really, really almost bland or somewhat flat. And so we were kind of wondering, like, why is that? Like, why does it look so flat? Does that mean it's not learning coevolution? And so this is where you might wonder why. And so what I'm trying to argue here, but before I go into more arguments, let me just explain what these concepts I'm referring to mean. So in a very, very simple model, what exactly is this input-output value that I'm plotting? What is this Jacobian is? And so essentially you could think of it as essentially some perturbation in the input and checking the perturbation in the output. And a Jacobian is essentially when you get to this limit of 0. So once you get to the limit of 0, it's whatever the slope of this line is, tells you what that gradient is and how the inputs to the output map. But what if you have a scenario where this model that you're evaluating doesn't actually have that nice linear effect or a nice curve, but instead it has a lot of flat areas, right? So like, what if I just made some input here, like a small little change here, and the output is exactly the same because you're in, so essentially this linear space, right? And so when we're computing a Jacobian over a protein language model, maybe the reason why it looks all flat because small little changes do not affect anything on the output. That's why it kind of looks flat. And so one thing we try to do is say, well, what if we actually make the step size larger? Like, what if we actually make a larger perturbation? So let's say if we want to see, like this right here doesn't change anything, but if we make this input a little bit larger, then maybe we'll actually start seeing differences in the outputs. And so we actually tried to do a scan of different step sizes, and probably not too surprisingly, we found the optimal step size was 1. And in hindsight, it kind of makes sense because 1 is roughly enough to maybe almost switch the category. In fact, if you maybe take a little bit larger step, you might completely flip the category within the model. And in practice, we find that actually just directly just changing the step altogether, like instead of just adding a 1, just replacing that with the 1-hot encoding of the other amino acid, it actually gives us better performance. And so every dot here is a different protein family, and I'm evaluating the contact accuracy. And so computing a Jacobian, which is essentially just a step size as a very, very small step size versus a step size of 1, got a huge boost. Now you actually see a very, very clear contact map. And finally, a step size of 1 versus actually just switching the category gives you a slight boost, but not too much of a boost. So this is now going to be our default protocol that we'll be following, and this is what we're going to essentially be evaluating for the rest of the talk is this 1-hot switch. So just to summarize this part of the talk, so the idea here is that you could take an input sequence, you could pass it to the model, get some kind of logits. These are the probabilities at every position for that amino acid. You can make a mutation at every position to all the other possible amino acids, and you get differences in the output logits. Now you can take the differences in the outputs and you get back this, what we for now are going to refer to as a categorical Jacobian. This is just a term that we made up, but if somebody has actually done this and gave it a different name, we're happy to change it as well. So anyway, so we're just calling it that for now. But the idea is like you could do all possible perturbations and get back a length by alphabet matrix. And just what I showed you guys with the Gremlin, I could also do the norm for every 20 by 20 matrix to get back a single value. And what this allows us to do, which is kind of cool, is to start to actually compare these values directly between the models. So we could take a multiple sequence alignment, in this case compute an inverse covariance, which is an approximation of Gremlin, then take the Jacobian over a protein language model and directly compare all the parameters to all the parameters. And one thing we see, the contact maps are very similar, but even the 20 by 20 matrices are actually very similar to each other. It's capturing similar probabilities for how the amino acids change to each other. And this is actually showing all the values, length by alphabet, and you can see that there's a reasonable correlation between the two. So this essentially tells us that it's not only that the context looks similar, but actually the parameters themselves that it's learning are actually almost identical between getting a multiple sequence. Now, the reason why they're not perfectly the same partially might be because maybe it's the MSA, like the implicit MSA that the language model is using, maybe includes more or less sequences. Like we don't really know what the actual MSA it's implicitly learning. And also potentially you could fill in information. Like there's a bunch of 0 values here that may be potentially filled in from other protein families. And so maybe you could do a better job at learning these parameters. Yep.
Can I ask a question? Okay.
Sure.
I just want to know if I correctly understood. So the Jacobian essentially is telling which output features are more sensitive essentially to change because it's where like the contact is happening. So this is like the intuition. Like I could use this to understand, for example, importance. Like these are the important. If it would be like an autoencoder or something, I could get the importance because that is where is the most important point in the output because a slight perturbation is affecting it the most.
That's a good question. So I guess you could call it importance, but I'm not sure if that's completely the way, at least the way, maybe the simpler way to think about it is like, let's say if position 1 is dependent on position 3.
Yes.
So if I mutate position 1, then I might have differences in position 3,
yeah,
and vice versa. And so you sort of get at least what, I guess you could say, the dependencies.
Yeah.
But whether or not that means importance is not always true. And what I mean, the reason why I'm saying that is because sometimes the most important things do not co-evolve. Like for example, let's say if you have a metal binding site, it might be completely conserved, and in fact, you won't see that in the matrix. And so I think the interesting thing about coevolution is that things that are actually co-evolving are actually not the most important things. They're sort of things that are medium important, because then they can tolerate substitutions.
And a related question was like, I think I might be wrong, but on computer vision, I read something about like the norm of the Jacobian is related to how much a point is out of distribution. So I don't know if the population you were mentioning before, like the rare proteins, have like a higher Jacobian norm because whatever you change is like you get something in the output that changes because the model is not getting what is happening, or something related. So there on vision was like if you are on the manifold of the images, then like it's challenging to get which point to change. But if you are out of it, like everything is like affected. So I'm going back to my because I come from different intuitions. I come back to this importance concept, but I don't know if this might be happening on.
I'm not sure. I'll think about it some more. Maybe we'll get back to you.
Okay.
Yeah. But that's an interesting connection. I haven't thought about it that way. Thank you. I mean, we're almost done with this section, but essentially, to summarize this, is like the contexts are similar, which is pretty cool, but also the parameters that we could extract from the models are similar to the ones that you could get from just training a simple model and that same protein family. And so this gives us a lot of confidence that, in fact, maybe the models are just indeed storing these coevolution matrices. And in fact, when you give it a sequence, it's essentially just extracting those same parameters. But there's still the possibility that maybe it's not doing a full fold lookup, right? Maybe it's sort of, you can imagine, like, if you were to compress all this data, like if you were to do it by hand potentially, you wouldn't go around storing a separate model for every protein family. You say, hey, these families both have helices. Maybe there's some shared set of parameters between these protein families. In fact, maybe you'd share like motifs, like how this interacts, and you store like mini gremlin models, right? And so Zidian in my group, who was visiting at the time, thought we would do a quick experiment to test for this. And so one idea is say, well, let's just say this is a little motif here. This is a local motif. We have a beta strand interacting with the helix. And what we could do is we could just mask everything except for that motif and ask the question, does the model understand that motif? And what understand means, does it have contacts that represent this interaction? And also for motifs that are separated from each other. For example, this helix separated by this helix, we can mask everything in between and on both sides and ask the question, how much contact is actually needed to recover those motifs? And so if it turns out you need the whole sequence, then we say, okay, the model does essentially a whole sequence lookup. But if it only requires that single small motif, then that tells us, okay, the model probably only needs that one segment in order to understand that interaction. And so here's a quick animation. And so here, what I'm showing here is literally taking this whole protein and masking everything except for that motif. And I think one kind of cool thing about this animation is essentially you can almost get rid of the whole protein, and there's only a certain part where essentially it starts to disappear. And so you can almost get rid of everything except for that motif, and you can see that it's still there. Now for the pair interaction, this one's actually kind of cool. It's like essentially you can almost get rid of everything except for that helix, and you could still see everything. So essentially all I'm doing here is removing everything from the sequence except for that motif, and then asking the question, does it understand the interactions between these two parts of the sequence? And it still does understand them. And so to us, this suggests that the model is indeed maybe learning not whole proteins, but learning motifs. And this could be a good thing for protein design. For example, let's say if you want to design a new protein, and you're like, hey, I want to recombine a whole bunch of motifs in different creative ways. This suggests maybe one way to be able to do that. That being said, it's also possible because it's just recombining motifs. The question is like, is it really novel, or is it just doing the same thing? So that's something up to debate. Okay, so this sort of leaves us with this hypothesis that it's essentially storing these motifs here. And so just to summarize this section, Jacobians are very useful because it allows you to analyze models, especially if you don't want to sort of think about what's going on in these black boxes. And we do have some links here for analyzing ESM models, for example, for analyzing protein and PNN models to extract contacts. But on top of that, you could also do this on every single layer. So this little animation here I'm showing here is computing Jacobian from input to layer 1 to layer 2 to layer 3. And you could sort of, the interesting thing is you could sort of see as you go through the layers, you start to build up more and more of the contacts, and eventually you get all the contacts on at the very end. So if you guys want to analyze this for your own model, check out the code here. And then finally for I think I only have 10 minutes left, right? Am I doing the math right? Here, okay. So finally for the last 10 minutes, I'll tell you guys about genomic language models, but not going to introduce any new concepts here. We're going to apply the exact same thing that I just told you about, but now to genomic language models. And one genomic model that you guys may have heard of already that was recently put out there is this new model called EVO. To me, this was actually an interesting model because they didn't use explicit attention layers. It was not one of those things that you could just look at the attention maps and ask the question, is learning contacts? You essentially were kind of forced to do this kind of Jacobian perturbation that I just told you about. So the idea here is that you have an actual DNA sequence, and the output is also a DNA sequence, and you can now perturb the input of the DNA sequence and ask the question, how do the probabilities of that sequence changes? So the exact same analysis, and there's a lot of crazy stuff happening in the middle, and I guess what I would say is like, let's just ignore what's happening in the middle, just focus how the inputs map to the outputs. And one exciting thing is if you give it an RNA sequence, you could actually recover all the stem pairings. And so you can get local contacts. At some point, I gave this talk at a different conference, and I said it didn't learn long-range contacts. Turns out there was a bug in the EVO code, and the authors emailed me and said they fixed the code. It should now recover long-range contacts, and it does indeed. So it turns out that they had some bug in how they, I guess, tile the hyena layers or something. But yeah, so now it's actually picking up these long-range contacts, which is quite exciting. But one thing, and also I just want to do a quick plug-in. There's another group that actually did something very, very similar to what we did, but more across all different DNA-based language models. And so if you guys want to read more about other models, you should check out this work as well. But for our purposes, we don't really care too much about the RNA. We care more about the protein side. So the question is, like, how about proteins? Do these models actually work on proteins? And so here is one particular protein family that has a huge amount of sequences. And so we don't doubt that it hasn't seen these proteins many, many times. And we see with protein language models, it actually captures the contacts really, really well. But genomic language model, where you essentially perturb the nucleotides and see how the changes in the outputs change, we see that it's actually very, very, once again, looks somewhat fuzzy. The reason why I'm showing only half of the matrix is because it is an autoregressive model. So essentially, when you perturb something in the input, everything changes downstream, but what you change downstream does not change what happens upstream. So so that creates essentially a half matrix. But for visualization purposes, you could always just drop it on the other side. One thing we try to do is say, well, maybe it doesn't understand proteins, but maybe it understands interaction of proteins, or maybe how the proteins are arranged in the genome. So if you give it, like, for example, an operon, in this case, 3 different proteins, it looks like it's learning some kind of splitting between them. There's these streaks that I think might be maybe related, like the start and stop codons. So maybe if you mess with the start codon, you probably start losing some information. But it doesn't seem to be understanding contacts in the same way that we have with proteins. And so this sort of leads to maybe two hypotheses about why it's not learning protein-protein side chain interactions. One, of course, scenario is like, well, now the task is much larger, right? You're trying to sort of store all the evolution of the genome. Maybe your capacity of your model is not large enough. You maybe just need to make these models larger, and that's what we saw with ESM models. Like, essentially, as you scale them, eventually they stored better and better contacts. Another potential scenario is maybe it's just the alphabet is wrong. And what I mean by that is often, especially for protein reasons, if you mutate one of the nucleotides, like for example, like the third codon or the third nucleotide of a codon, you're really easy to recover what that amino acid is without even thinking about what the rest of the sequence is. So maybe the way the model is trained by just masking individual nucleotides, the model never had an incentive to learn these long-range protein-protein interactions. Luckily for us, in collaboration with Yuna and Andre from the Tata Bio company, they actually said, well, you know what if we actually train a new language model, but in this case, we purposely use just amino acid encoding for the amino acid positions, but then use this BPE encoding for the rest of the DNA parts. So the idea here is that you could take a chunk of the genome, in this case a metagenomic context. For parts that are protein coding, you actually encode with an actual amino acid. So you have 20 tokens. And parts that are DNA or non-coding, you can just encode with these little segments. And these segments could be of variable lengths. And so then during training, you can actually mask entire, effectively, codons instead of masking individual nucleotides. Maybe another way of we could have done this is to say, well, let's actually encode like the 64 codon tokens. That could have been maybe a better way to do it. But in this case, encoding the individual amino acids as essentially, I guess every codon to an amino acid token. And so when you do that, and then you analyze, for example, protein-protein interactions. So in this case, this is one protein ABC transporter interact with another protein. If you run these through Gremlin, we get contacts. That's not too surprising. These things are co-evolving with each other. And we see the same thing with genome language models. Like we can do mutations in protein A, and we see that the logits in protein B actually change as well. So you actually see the interactions appearing in those guys. Just to contrast that with the two other models. So this is the EVO model. We see nothing there. It's like just blank, not learning anything about the interaction. And the same thing with ESM too. Like you give it the two proteins, and it doesn't give you back any contacts between the two proteins. I mean, it's never seen those two proteins together, and so that's not too surprising. But I think it does definitely, like I guess the summary here is that the tokenization does definitely probably play a big role. Maybe not necessarily tokenization, but how you mask. So maybe like for protein regions, maybe it makes sense to mask like 3 nucleotides at a time that are close together. Maybe for non-coding regions, you want to mask individual nucleotides. Otherwise, you might not get enough resolution for DNA strands and so on. So I think it's something to keep in mind when we're training these models. OK, so with that, I think I'll stop. Some people to thank. So I think I described a couple of projects here today, and so there's a lot of people that were involved throughout this whole thing. But if anybody's interested in collaborating, maybe interested in joining my group, I just started up at MIT in January, so we're relatively new. And so if anybody's curious, feel free to stop by, say hi. All right, well, thank you very much. I'll stop there. Yep.
Thank you for this wonderful talk. So I had a question. The way I understood the categorical Jacobian method is that it's sort of learning a general covariance matrix. And I guess something I was wondering about, do you think there is something special that these PLMs learn so much about protein structure and that it correlates so well to this categorical Jacobian that you compute? Like, could it be learning that there are some other properties that the categorical Jacobian is capturing? Like maybe a two examples that you have two complementary, some sort of active sites that have common function and they don't necessarily interact with one another, but the model learns that they're codependent. Would it be captured in this Jacobian, but maybe not necessarily be reflective of structure?
I mean, in some ways, we are perturbing pairs of interactions. And so I guess if one wanted to go crazy, you can compute like a Hessian or something, where you can maybe get some higher order stuff going on. Yeah. So in this case, we're explicitly perturbing pairs of things. And we do see the highly correlation with structure, but I guess one thing I should mention is the correlation is not 100%. It ranges from like 50 to 90% across different proteins. And it might actually be interesting to look at those examples where it's like 50% accurate. Maybe it is actually picking up on things that isn't just protein.
Maybe these like genomic models, they're learning something else. That's something that's beside.
Great question. So like in this case, like it does feel like it's learning maybe the starts and ends of genes. But the reason why it's not learning protein contacts, I suspect it's mostly just because of how it was trained. Just because they mask individual nucleotides instead of trying to mask individual codons. So I think if they retrain it with just masking of codons, I think it would start to pick up on interactions. Unless that's actually what you want. Maybe you purposely want it to have two models. One learns protein, one learns gene structure or something, and then maybe that's a good way to disentangle those signals. Yeah.
Put up an example of the contact matrices learned by Gremlin and the protein language model, and there's correlation was 0.6 or something. I am really curious which one is more accurate just on this particular example.
That's a great question. So the accuracy of the contacts are actually more accurate for the protein language model. So like if I compare the ground truth contacts coming from the crystal structure, they are more accurate. So I guess what I'm saying is like we're not expecting it to be one because we expect the protein language models to get better, just because they're able to maybe use more information or maybe filter information in different ways. One of the sort of still an unsolved problem in the MSA space, so people who work on multiple sequence alignments, is like questions like which sequence should I include and not include? And then also like how do I align these sequences to each other? And so we would expect a model that was not explicitly forced to do either of those tasks may have learned a better version of that to some extent.
And I have another question that's maybe related, but why? What's your intuition for why in the protein language model it only learns the contact map in the final layer, whereas in the other model it's in the first and only layer of the model? Like, why does it take so many layers?
Oh, which other model? Maybe.
Like just the Gremlin model, right?
Oh yeah. Well, I think the key part here is that if you have if you're just training on a single protein family, you don't really need that many layers. Like we actually tried many layers, and we found that you could just go to one layer and still get similar pseudo likelihoods. And so I think the reason why that's true is because go back a little bit to the slide that I had. Essentially, all the parameters of the coevolution could be literally just stored in the queries and keys. And just to go back to this picture here, like for example, ultimately this is what we're trying to learn. And a single layer already has those sets of parameters. Like both the queries and keys could be already represented in just a single layer. Now, of course, if you're training across many, many different protein families, then maybe you need to have multiple layers for like some kind of lookup algorithm. So I guess if we believe what I'm saying, that ultimately protein language models are some kind of lookup algorithm, then maybe what's happening is like the first few layers are saying, okay, which motifs do I see here? I need to look them up and stitch them all together. And by the time you get to the end, you finally put them all together. One little side note here, the genome language model folks also found that sometimes like the middle layers are much better for homology search versus the last layers. Like so for example, if you take the representations at different layers and ask the question, which one will find the homolog best, it turns out some of the middle layers actually do better job at that. And the reason I'm bringing this up is because I feel like that adds some evidence that maybe, I guess you could say the beginning and middle layers are essentially looking things up. And so if you also want to look up things, this is probably the best representation to be using. But if you're interested in getting contacts, maybe the last representation is what you should be using. And so I guess different parts of this. I guess.
Thanks, Sergey, for the great talk. I had a question related to your three panels where you're showing folding versus lookup versus motifs. So there are many cases, I think, where we want to learn folding. We want those scaling laws across protein families to be straight lines because that allows us to generalize to more de novo settings and explore a broader part of the protein space. And so I'm wondering if you've had any ideas of how to factor out this information from the models so that the model factor out coevolutionary information so that models learn more physics-based folding. Just do you have any ideas of like basically how to avoid cheating on coevolutionary information?
Yeah, I'm not sure if there's an easy way to do that. I mean, one thing we assume is happening is like as you start training the model. So let's say you take a sequence, you mask a position, and you want to recover that position. Like the simplest thing you could do is say, what does this sequence belong to? Like what family does it belong to? So there's probably some version where it's like learning some kind of k-means or some kind of clustering, and you can return that probability. Then as your model gets larger and larger, you say, well, besides just learning like the means of each matrix, you could also start to learn the covariance of each matrix. And maybe as your model gets larger and larger, you could probably learn some trivariance. I don't know if that's a thing, but you could essentially start to maybe learn higher order things. But I don't see at what point will it decide, hey, it's time to not just store the statistics of these families, and when do I start to try to reason why those statistics exist? Yeah, I don't have a good idea of how to make it go beyond that. And does anybody else have any ideas how to push the models to snap out of conservation coevolution?
If you add like the mean and the covariance as input to the model as well, and try to, so it doesn't have to learn it. I don't know.
Oh yeah. So you're saying maybe we can freeze this model and then learn like an offset to the model to some extent? Yeah. That's a good idea. All right, next time you could present on it, I guess, see if it works. Yeah, but that's an interesting idea. Yeah.
Yeah.
All right, thank you so much, you guys.
Thank you.